Here we'll look at how to go about doing dihybrid crosses. Now, in a dihybrid cross, as the name suggests, you're looking or following two genes. So di meaning two. You're going to follow the outcome of crossing individuals, and you're following the pathway of two genes through the cross rather than one. So let's look at an example. Let's take the hypothetical example of the colour and the shape of pea seeds. Alright, so here we're going to dealing with two genes. So one gene will be for the colour of peas, of which there are two alleles, yellow and green. Yellow will make yellow dominant, so yellow peas will be controlled by the big Y allele, and green peas by the little y, it's recessive. Now, just a little note here, when you're using the same letter for dominant and recessive alleles, make sure that you clearly distinguish for the examiner uppercase and lowercase lettering, otherwise it becomes very confusing. To do with the shape of the peas, let's go round peas. We'll make dominant, so we'll make that capital R, and wrinkled peas will be recessive, and that will be little r. So let's say we have parental cross between two individuals, one with homozygous dominant yellow peas with homozygous dominant round, crossed with homozygous recessive green and homozygous recessive wrinkled. So the parental genotypes big Y, big R, crossed with little y, little y, little r, little r. Now the important thing to realise here is that each of these genes is on a separate homologous pair. So it's important to be able to visualise what's going on. So on one homologous pair of chromosomes will be the Y gene for colour, and on the another pair of homologous chromosomes there will be the gene for seed shape. Over here we would have little y, and little r on a separate homologous pair. So it's important to be able to visualise this. The two genes are on separate homologous pairs of chromosomes. What happens to each member of one homologous pair during meiosis, remember, because these separate into different gametes, doesn't influence what happens to the other pair. These separate into different gametes. So let's just have a look at how this might work in practice. So this chromosome carrying that allele could end up with that chromosome in a gamete, or this chromosome here could end up with that chromosome in a gamete, or this chromosome could end up with this chromosome in a gamete, or this chromosome could end up with that chromosome in a gamete. So they are the four possible combinations of the chromosomes ending up together in the gametes, and likewise for this example here. So let's have a look at what that looks like. So we have Y, big R, crossed with little y, little r. This is the parents. So the possible gametes, in this case all the gametes will contain a big Y and a big R. So if this one ends up with that one, it'll be big Y, big R. If this one ends up with that one, it'll be big Y, big R. If this one ends up with that one, it'll be big Y, big R. If this one ends up with that one, it'll be big Y, big R. So all the gametes will contain big Y, big R. And over here, likewise, all the gametes of this plant individual will be little y, little r, for the same reason. So there's only one possible way that these can come together in fertilisation. So if any gamete from this plant meets any gamete from this plant, then all of the F1, the first generation offspring, will be big Y, big R, little r. Alright, so they will be yellow, and round seeds, 100%, and 100% genotype. Now let's see what happens if we cross two members of the F1 with each other to generate a second generation of plants. So here we're going to cross a big Y, big R, little r individual with a big Y, big R, little r individual. So this is the F1 parents. Now let's have a look at what happens. Now in the gametes, remember, this chromosome carrying big Y could end up with this chromosome carrying big R in gametes. This chromosome carrying big Y could end up with the chromosome carrying little r, so we could end up with gametes with that combination. This chromosome with little y could end up with this chromosome with big R, or this chromosome carrying little y could end up with this chromosome carrying little r. So 1/4 of the gametes will contain that combination, 1/4 will contain that combination, 1/4 will contain that combination, and 1/4 will contain that combination. And of course it's exactly the same for this individual over here. So 1/4 of the gametes will be big Y, big R, 1/4 will be big Y, little r, 1/4 will be little y, big R, and 1/4 will be little y, little r. Now this is where setting up a detailed Punnett square is important, and this will be a 4x4 Punnett square because each individual has 4 possible gametes. So big Y, big R, big Y, little r, little y, big R, little y, little r, big Y, big R, big Y, little r, whoops, little y, big R, and little y, little r, making sure your letters are clearly distinguishable. So let's have a look at what the F2, the second generation, possibilities are for the offspring. So we can end up with big Y, bigger R, big R, big Y, big R, little r, big Y, little y, big R, big R, big Y, little y, big R, little r, big Y, big R, little r, big Y, little r, little r, big Y, little y, big R, little r, big Y, little y, little r, little r, big Y, little y, big R. You'll notice that I tend to put the dominant allele first. It's just a matter of convention, but it really doesn't matter what order you put them in. So big Y, big R, little r, little y, big R, little y, big R, little r, big Y, big R, little r, big Y, little r, little r, little y, big R, little r, and little y, little r, little r. So they are the 16 possible genotypes in the F2 offspring. To do a bit of analysis of this, you've got to work out the number of different phenotypes that come out of it. So let's have a look at how many yellow round seeds would there'd be. So anything with a big Y, big R will be yellow round. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 out of the 16 combinations will give you a yellow pea and yellow round pea. So 9 will be yellow round. Let's take a different colour. Let's go red to see how many will be yellow wrinkled. That'd be a big Y and then 2 little r's for wrinkled. So this would give you yellow and round. This would give you yellow and round. This would give you yellow and round. And that's the only possibilities. So 3 will be yellow round. Sorry, yellow wrinkled. My apologies. Yellow wrinkled. Let's have a look at how many would give us green and round. Let's go a different colour again. So green would have to be little y, and round has to have at least one big R. So this would be green and round. This would be green and round. This would be green and round. So we have 3, which would be green and round. And you'll notice we have 1 left over, which will be green and wrinkled. So 1 will be green and wrinkled. So in a cross between 2 heterozygous parents, we end up with a ratio of 9 is to 3 is to 3 is to 1 of the phenotypes. And this is a classic ratio for a cross between 2 heterozygous parents.